Welcome back to Dr. Liu's VCE Biology. Today's topic is human change over time and scientific investigation, as presented in the 2025 NHT Biology exam. Let's learn together. If teachers, parents, or students notice mistakes in my explanations, please provide me with your feedback. The chimpanzee-human divergence data is estimated at between 8 and 5 million years ago. An unclassified hominoid skull was discovered in Africa, dated at 7.2 to 6.8 million years old. The images show the underside of the skulls of a chimpanzee, Pantroglodytes, Australopithecus afarensis, dated at 3.7 to 3 million years old, the modern human, Homo sapiens, and the unclassified skull hominoid. The arrows point to the foramen magnum on each skull. Analyzing the foramen magnum is the opening at the base of the skull where the spinal cord connects to the brain. Its position is a primary indicator of whether a species walked on two legs, bipedal, or quadrupedal. Pantroglodytes, chimpanzee. The arrow shows the opening is positioned toward the back of the skull. This allows the head to face forward while the body is horizontal or tilted. Homo sapiens, modern human. The opening is positioned centrally, toward the front middle of the base. This balances the head directly atop a vertical spine. The unclassified skull. If you look at the position of the arrow on the unclassified skull, the foramen magnum is shifted significantly forward compared to the chimpanzee. It aligns much more closely with the centralized positions seen in A. afarensis and H. sapiens. Evaluating the options. A is incorrect. Age alone does not determine locomotion, anatomical structures do. B is correct. The forward, anterior, shift of the foramen magnum is a classic evolutionary adaptation for bipedalism, as it allows the skull to sit level on a vertical vertebral column. C is incorrect. Visually, the position does align more closely with the bipedal specimens than with the chimpanzee. D is incorrect. This option contradicts the visual evidence. The foramen magnum is clearly not at the extreme back of the skull like the chimpanzees. Scientists found fossilized teeth and fossilized humerus, upper arm, bone in Indonesia. The fossilized remains belong to an unknown hominin species, species X. The fossils were dated at around 700,000 years old. To determine the evolutionary relationship between species X and other hominin species, the teeth and humans were compared to those of three other species: Homo erectus, Homo floresiensis, and Homo sapiens. All species lived in Indonesia. The results are shown in the table below. The correct answer is A. Rationale for A. Species X, 700,000 years old, lived after H. erectus, 900,000 years old, and share similar teeth. This chronological order and morphological similarity suggest a potential ancestral relationship. Why others are incorrect? B. A species is generally not considered a putative species of two distinct species simultaneously in this context. It is usually an ancestor, descendant, or sister taxon. C. Living in the same location, Indonesia, does not make two groups the same species, especially when they lived 640,000 years apart, 700,000 vs 60,000 years ago. D. H. sapiens is the least similar to species X, with both teeth and humerus listed as different. Rationale for D. The fossils are dated at 700,000 years old. At this age, direct dating of the fossil itself is difficult, so scientists typically use stratigraphy or radiometric dating of the volcanic rock layers, such as argon-argon dating, in which the fossil was found. Why others are incorrect? A. Carbon-14 dating is only effective for organic samples up to approximately 50,000 years old. It cannot date a 700,000-year-old fossil. B. DNA degrades over time and is extremely unlikely to be preserved in a 700,000-year-old specimen, especially in a tropical climate like Indonesia. DNA sequencing is a tool for identification, not a primary dating method. C. Comparing them to H. fluoresciences would be inaccurate for relative dating because H. fluoresciences, 60,000 years old, is much younger than species X. The EPDR1 gene found on chromosome 7 helps control the contraction of muscle in the hand of humans. This gene has many alleles. Some of the alleles have a Neanderthal origin. A Homo neanderthalensis fossil was located in Europe and dated to around 120,000 years ago. The scientists discovered that the genome had an identical EPDR1 allele to that found in some individuals currently living in Europe. The correct answer is B. Rationale. The discovery of an identical EPDR1 allele in both a 120,000-year-old Neanderthal and modern Europeans indicates interbreeding. When two different species, or subspecies, mate and produce offspring, alleles from one population are introduced into the genome of the other, a process known as gene flow. Why A is incorrect. If they were entirely different species with no gene flow, they would be very unlikely to share an identical allele sequence for this specific gene. Why C is incorrect. While mutations happen, finding an identical complex allele in two different groups is much more likely to be the result of shared ancestry or interbreeding than a coincidental identical mutation. Why D is incorrect. The evidence of an allele in Europe does not prove a migration pattern from Europe to Africa. If anything, fossil records generally show the genus Homo moving out of Africa. DNA sequencing of the human Y chromosome in individuals can provide evidence for the migration of human populations around the world. In 2002, scientists recorded the numbers of short tandem repeats, SDRs, on a section of the Y chromosome from individuals in South Asia and Aboriginal Australians. The scientists found similar SDR variation between the groups, with divergences from a recent common ancestor dated at around 5,000 years ago. In 2016, a repeat study was undertaken using improved DNA sequencing techniques and extended to include Papua New Guineans. Scientists constructed a phylogenetic tree from the results of the 2016 study. The correct answer is D. Rationale. The 2016 phylogenetic tree shows that the common ancestor for Aboriginal Australians and South Asians dates back to approximately 54,000 years ago. This directly contradicts the 2002 study, which suggested a much more recent divergence of around 5,000 years ago. Why others are incorrect. A. Close relatedness is determined by the recency of the common ancestor, not just the number of divergences. B. While they diverged long ago, divergence time alone is not the sole measure of comparative genetic diversity between the two groups. C. The tree shows branching within the South Asian group around 27,000 years ago, but there is no evidence that this resulted in a new species of Homo sapiens. The correct answer is C. Rationale. The text states that the 2016 study was a repeat study undertaken using improved DNA sequencing techniques. In science, using more advanced technology and a larger data set, including Papua New Guineans, generally leads to results that are considered more accurate and reliable than earlier, more limited studies. Why others are incorrect. A. Scientists frequently repeat studies. Having different researchers perform the work does not inherently make a study imprecise. B. Recording different types of data, SDRs versus constructing a tree, does not make the studies invalid. they are simply different ways of analyzing genetic relationships. D. Science is iterative. True values are often refined as better technology becomes available, rather than being permanently settled by the first study conducted. A recent study summarized the trend in brain size for Homo species over time. Data was obtained from 19 different studies over 90 years. The results are presented below with brain size measured as cranial capacity. Analysis from H. habilis to H. sapiens, 300,000 to 129,000 Ya. There is a significant increase in cranial capacity. Data points. Brain size grows from 678 cubic centimeters in H. habilis to 1,464 cubic centimeters in H. sapiens, 300,000 to 129,000 Ya, more than doubling in volume. Advantage: larger brain size, increased cranial capacity, generally allows for increased cognitive function and complex social behaviors. Details: this includes the development of advanced tool-making, language, abstract thought, and improved problem-solving skills, which significantly enhance survival and adaptability. While the general evolutionary trend is toward larger brains, the data shows a recent decrease in cranial capacity in modern humans. Data: cranial capacity peaked at 1,464 cubic centimeters, 300,000 to 129,000 Ya, and has since declined to 1,304 cubic centimeters in present-day H. sapiens. To improve accuracy, scientists could increase the sample size by including data from more fossil specimens or diverse global populations to ensure the averages are representative of the entire species at those times. Anatomical and fossil evidence. Locomotion, bipedalism. The position of the foramen magnum, the opening for the spinal cord, is the primary indicator of bipedalism. A centrally positioned opening, anterior shift, indicates bipedalism, while a rear-positioned opening, posterior, indicates quadrupedalism. Case study: species X, a 700,000-year-old Indonesian hominin likely evolved from H. erectus because they share similar dental features and follow a logical chronological sequence. Dating techniques: carbon-14 dating is only accurate for samples younger than 50,000 years. For fossils aged 700,000 years or more, scientists must use absolute dating of the surrounding volcanic rock layers. Genetics and migration. Interbreeding: the presence of an identical EPDR1 allele in both Neanderthals and modern Europeans serves as definitive evidence of gene flow. Interbreeding between H. neanderthalensis and H. sapiens. DNA phylogeny: genomic studies often require revision as technology improves. A 2002 study estimated a split between South Asians and Aboriginal Australians at 5,000 years ago. A 2016 study using improved sequencing corrected this, showing the divergence occurred approximately 54,000 years ago. Evolutionary trends in brain size. Growth trend: from H. habilis to early H. sapiens, cranial capacity more than doubled, from 678 cubic centimeters to 1,464 cubic centimeters, providing a significant advantage in cognitive function, social complexity, and tool use. Modern decline: contrary to the assumption of constant growth, modern human brain size has decreased from its peak to an average of 1,304 cubic centimeters. Data integrity: to improve these findings, researchers suggest increasing sample sizes and including more geographically diverse fossils to ensure the data is truly representative. If you need my help, please add me on WeChat or WhatsApp and send me an email. Thanks for your attention. Please note Dr. Liu's VCE Physics channel, Dr. Liu's VCE Chemistry channel, and Dr. Liu's VCE Biology channel.